Biology can be defined as the scientific study of life. And what makes biology scientific is that it is not enough to go around making observations and describing the things you see and then making a guess of what it is that you may be observing without doing an investigation, without actually following a proper procedure for discovering exactly what may be taking place before your eyes. And so in this presentation, we're going to explore exactly what is this process of science and what makes biology a scientific field. There are two main forms of investigations biologists can do. One of those is going to be known as discovery science. Discovery science involves going sometimes to a natural setting, like you see. In the case of this photo, there is a submersible type of vehicle that can go into the depths of the ocean to not only take images of what they see, but also capture samples of what lives deeper in the ocean. Through a lot of data collection, through a lot of careful observations and meticulous detailing of everything in the surroundings, scientists, like marine biologists, can discover more about life underneath the sea. Another type of a scientific investigation can be what scientists do in a laboratory, and they are studying the genetic makeup of organisms, including the genetic makeup of humans. By now, most people know that the genome describes the genetic makeup, that is the entire collection of genes that can be found in the DNA of an organism. And that is another example of discovery science. Notice that there is not necessarily an experiment with controlled variables that are being used in that case. It is simply about very carefully recording, detailing everything that is observed in all of the natural surroundings. Another type of investigation is going to be hypothesis-driven science. And this is how most of the biology concepts in your textbook have come about. In hypothesis-driven science, a scientist will be in the business of formulating hypotheses. A hypothesis can be defined as a tentative explanation for a specific phenomenon one has observed. So notice that I'm using the word tentative. Tentative means that we don't know if the explanation is actually the correct explanation for what we're seeing. You would only know if the hypothesis is supported, if it's telling you something true about what you're seeing, if you test it. That is the second main step in a scientific investigation. First, you formulate a hypothesis. It is tentative. Number two, you test it to see if there's merit, to see if there's any validity in what this hypothesis is explaining. Notice that the process of science is going to be a deductive form of reasoning. A scientist is going to try to study something very specific, and from that hypothesis, which may be supported, make broader explanations, which eventually can be described as a theory. Take a look, for example, at this illustration, and you can see from the sequence of events in the scientific method that the scientific method really isn't something strange or bizarre. It is actually how we go about solving everyday problems. The first step in the scientific investigation is going to be observation. You may find yourself hiking in your favorite forest and you notice something you haven't seen before, and there's also a context that may be explaining what you see. Those careful observations sometimes lead to questions. What is happening with that plant, with that animal you're observing? Much in the same way you can solve something trivial, like in the case of this slide here in this illustration, somebody is trying to use a flashlight, and their observation when they turn on the flashlight is that it doesn't produce any light. Observation: the flashlight doesn't work. Question: what is going on? What is the reason for the flashlight not to work? And then you begin to think of all of those possible explanations that may be the reason behind this flashlight not working. And you know that flashlights require batteries. You know that flashlights also rely on a little light bulb that will be producing the light. And maybe there is a switch that is problematic. And you can come up with any one of those possible explanations and make that your hypothesis. So let's say, for example, that you follow the hypothesis that the batteries are dead. You can make a prediction. The prediction is, if I replace the batteries, then the flashlight will work. Predictions usually follow that type of pattern. If, then. Now, the batteries can be replaced. The prediction is tested. Now the flashlight has new batteries, and it turns out that nothing happens. What happened with the hypothesis? You can say from a statistical point of view that the hypothesis is rejected, but the hypothesis is voided. It didn't provide the best explanation for what phenomenon you had, which is the flashlight did not work. Or you may go with a different hypothesis. The hypothesis that maybe the bulb has burned out, that you can come up with a new prediction. If the light bulb is burned out, then replacing the light bulb and putting a new one is going to make the flashlight work. You do that work, you test the prediction, you do the experiment, and when you turn on the flashlight again, there it is, the flashlight works again. One important thing that I want you to know about hypotheses is that they must be falsifiable. This tentative explanation you're providing, there must be a way that you can test it and it will either be supported or it's going to be rejected. Another detail is that a hypothesis that is falsified, meaning a hypothesis that is not supported, is not a waste of time. It is not a failure, because in the end, you have gained knowledge that you didn't have before. For example, in this case, the batteries didn't solve the problem. You may be disappointed, but it is not a waste of time, because now you know something that is not the cause for the flashlight not to work. Likewise, in a natural setting, scientists go about making careful observations, making predictions, formulating hypotheses, testing those hypotheses, and analyzing the data. In your textbook, you will find this elegant and simple example of scientific inquiry at work. It comes from a study of mice conducted in the panhandle of Florida. There you can find some white beaches, and living along the grass and the dunes, you're going to find mice that are going to have a light coloration, and they blend real nicely with the sand. A person with a basic knowledge of biology may say, oh, look, the mouse is camouflaged, and that camouflage can be a good strategy for avoiding a predator. Then you move about 30 kilometers, that is about 20 miles inland, and the environment is different. There is a darker ground with darker vegetation, and you find that for the same species, it's the same type of mouse, same species, the fur of those mice living in that darker place is going to be darker, once again, making those mice camouflage, hide from the detection of predators. And so you can't just go and say, well, look, these mice, depending on where they live, are camouflaged, light for mice living in the light color sand, darker for mice living in the darker vegetation inland, and take it as for granted. You can't do that. In biology, you have to follow the process of science. You have to go and follow that observation that, hey, there seems to be some camouflage with your hypothesis. The hypothesis here is that fur coloration is an adaptation, is something that has evolved as a strategy for avoiding predators. What do you do next? You come up with your prediction. If light colored mice are indeed camouflaged when they live in the light colored sand, then predators are not going to be attacking as often those mice. And you can develop a similar prediction for the other case, for the mice that live inland. A person by the last name of Hopi Hofstra, decided to do an experiment and verify this explanation, this hypothesis, to see if the hypothesis should be falsified or if it is supported. And what she did with her collaborators is that they made up these mouse models. They look like a real mouse, but they are not. And they placed in the beach habitat where the sand is light colored, some models with the light fur and some models with the dark fur. They left them there and then they returned later to see which ones of those mice had been attacked or had been taken away by a predator. Big disappointed for the predator, right? And so what they found out is that those light colored models, the mice, that remember, they're not the real thing, it's just a model, were only attacked or removed by a predator 25% of the time. But in the same light colored sand, the dark mice were attacked or picked up by a predator at the rate of 75%, 3 times greater rate of predation or attacks for the dark mice compared to the light mice in the light colored sand. Now you have done a test. Now you have provided evidence that the hypothesis of camouflage as something that has evolved as an anti-predator mechanism for these mice has validity. Well, you can replicate, do the same experiment inland with similar results. Light fur mice in this darker background are going to be attacked at a greater frequency than dark fur mice against this darker background. Now, when you have not one, but actually 2 replicates, 2 studies in exploring the same hypothesis, but in different places, confirming the same explanation, now you can say that your hypothesis is supported. When you're doing controlled experiments, it is also important to recognize that there are variables. In the case of this study, the color of the mice is going to be the independent variable, one that the experimenters are manipulating and selecting which models to be placed in what particular background. The rate of attack is something that depends on this independent variable. When a hypothesis is supported and you do a replicate of the experiment with a similar type of a species in a different kind of background, and time again, that hypothesis is supported, it can give rise to a theory. Theories in biology are not just going to be any guess. Like when you see somebody on a TV show at a crime scene and they just arrived and they say, well, I have a theory of what happened here. No, they don't have a scientific theory. Maybe a theory in the area of the kind of job that they do. But in science, a theory is going to be something that is supported by many hypotheses that have been tested and provide evidence of the same kind of an explanation. And so in this case, the study of the fur mouse, called the mouse fur color, giving the same kind of result can be used for adding evidence to the evolutionary adaptations theory arising by the process of natural selection. Theories are going to be explanations that are broad in scope. As I said, they are supported by a large body of evidence. And sometimes from theories, you can go to a greater level of explanation, which is going to be putting them in a form that can be tangible. Something intangible may be easier to be understood when you have a visualized form of understanding nature, and that's called a model. And so here you see, for example, models of cells. Here we have a eukaryotic cell, of which you're learning also in this introduction to biology. And here's going to be a prokaryotic cell, smaller for the prokaryotic, larger for eukaryotic. The eukaryotic has many organelles. You don't see very many organelles here in the prokaryotic cell. Eukaryotic has a nucleus. Prokaryotic cells don't have a nucleus. And so these models can be representations helping us understand better theories like the cell theory we use in biology. And so that is going to be all for introducing you to the process of science and how scientists go about exploring life in this planet.